So hello and welcome to the Sheekey Science Show, where today I speak with David Meyer, an aging researcher and bioinformatician at the University of Cologne. David's work revolves around the use of aging clocks, either through DNA methylation or transcriptomic data, to better understand the aging process. Here we discuss his research, in addition to his thoughts on programmatic aging versus stochastic aging. I hope you enjoy our conversation. And maybe we should backtrack a little bit for our audience and just actually try and explain what aging clocks are.
Mm-hmm. Of course. So aging has been tried to be measured already for some decades. Most popular and maybe also then popularizing the field was Steve Horvath's pan tissue clock, which he published roughly 12 years ago now, I think, where he used again these DNA methylation sites and tried to use these to find pattern in the data to predict the chronological age of a species. So how this is done is he takes a lot of data, a lot of DNA methylation data from a lot of cohorts, so with an age range from, let's say, young to an old human. And then he uses machine learning. It's often something as simple as a linear regression with a few extra steps maybe, that then try to find the pattern in the data. So of course, if you have a CpG site that goes up linear with age, that alone is already a pretty good indicator. So if you would measure this single CpG site, you could of course already get an idea of how old a person is. But every single CpG site on its own is not really that informative, so you usually combine a lot of CpG sites to get a more robust clock. Although it has been shown that 3 CpG sites alone are already sufficient to predict the chronological age to a pretty high accuracy. And this now has been extended to also other data sets and to also try to not predict the chronological age, but maybe more biological readouts like mortality directly. You can go into more detail if you want, but I guess that's the main idea of aging clocks.
Yeah, exactly. And you already mentioned this sort of, to me, it's always very surprising that just 3 of these sites can actually be used to predict someone's age. And in age, are we talking about chronological age? Are we talking about biological age?
Mm-hmm. So initially, we used the aging clocks were built on chronological age, essentially also due to a limitation of data. So chronological age is much more available. And this works really well. So now we can build chronological aging clocks that really predict the age of an individual with ever of around, let's say, 2 years on average. So they are highly accurate. Now the problem here is, of course, that if you would have a perfect chronological age predictor, then it wouldn't tell you anything interesting except your age, which you could just get from looking at the passport, essentially.
True.
So the idea of this, we call it first generation aging clocks that train on chronological age, is that you need a prediction error. So you need a variance around the predicted age, chronological age, sorry. And that the hope is that if you're predicted older than your chronological age, that this then indicates that you are aging faster than the average person. So, and this has been shown that often this is actually associated with mortality or certain diseases. However, the association is not as strong as you maybe would wish it to be, which is why in a second step, so in a second generation of aging clocks, people have not used chronological age to train the clocks, but, for example, mortality. This data is much more limited. But if you train on mortality, for example, this is, of course, directly a biological readout. And this then also tracks much better with certain diseases and mortality in general. So these are much better predictors of this biological information.
I see. And I think perhaps you've answered another question that I have, which is, why are there so many clocks? Even just if we talk about DNA methylation, there are so many clocks, and I just generally cannot understand why there are so many.
Yeah, that's an interesting point that you mentioned. So I think initially with Steve Horvath's pan tissue clock, what he showed is that you can use one single DNA methylation clock for humans that can predict the age of a variety of tissues. Now, what he showed, though, is that not every tissue works equally well. Some tissues just work better than others, potentially also because for some tissues we don't have or had that much data available. So what people then afterwards also did is to train chronological aging clocks on specific tissues with more data to get a more accurate predictor for specific tissue types. And then subsequently also to not only predict chronological age, but as I mentioned, to have more biological readout like mortality directly. And now in the last step, some people call it third generation aging clocks, to not only have a pan-tissue clock, but a pan-species clock, like for example the pan-mammalian clock that came out I think 2 years ago now, where they could build one single clock that tracks the biological age of a variety of mammals, which is of course remarkable that it works at all. So I think sometimes if you have a rare data set, a rare tissue type species, you might need to refine your clock and build your own clock to be able to do that. And that is probably why so many clocks are popping up at the moment.
Yeah, no, that does help a lot more with my understanding. But I think the other thing, I mean, specifically with the DNA methylation clocks, is what does that actually tell us about the aging process? Like, are these marks just correlating, or are they telling us something about the cause of the aging process?
Mm-hmm. So DNA methylation generally is a bit harder to interpret, I would say. So if you would have gene expression or protein levels, then you could directly see how certain genes or proteins go up or down with aging. With DNA methylation is of course much more difficult because you can have cis or trans effects, and often you have of course multiple DNA methylation sites in the vicinity of several genes. So it's not as easy to directly predict the consequences of changes in DNA methylation, although there's of course progress also. However, there are still some pathways that seem to pop up quite often in the DNA methylation clock field. So for example, often developmental processes, so genes that are involved in developmental processes, seem to be enriched in the CpG sites that make up the clocks. And of course, this has then led to ideas that maybe there's a programmatic aspect of aging, that the developmental processes just continue running throughout the aging process and maybe drive aging. However, what we now think is that is not directly the case. So last year we have published this paper, Nature Aging, where we showed that what aging clocks do seem to measure is actually what we call accumulating stochastic variation. What do we mean by that? So we essentially mean with stochastic variation how noisy your data gets. So if you're young, everything is well regulated, everything is happening as it should be, and genes that are supposed to be active are active, and genes that are supposed to be not active are, well, not active. So for example, if you have a skin cell, you are expected to have a skin program, so the genes and proteins important for the skin are active, but you don't need all the genes that are important for liver cell or brain cell. These are suppressed. But now, with aging, as I also briefly explained before, you have this accumulation of errors. So the DNA methylation sites that are usually there to repress certain genes just randomly get removed or just not copied. And over time, this then, of course, trickles down, and you have a reactivation of genes that are not supposed to be active, or maybe a repression of genes that are supposed to be active. So just by random chance, essentially. And if you now think about developmental processes, these are, well, important for development. So after development ends, these are repressed. And if you have now a 100% repressed gene, just by random chance now, of course, this gene is slowly getting reactivated over aging. So it's getting de-repressed. It was completely repressed, but now with these errors accumulating, what we call accumulating stochastic variation, you have more and more gene expression re-emerging. And this is also what you see in biology. So you see that developmental genes are getting expressed again, or you see a loss of cell identity, because also genes that are supposed to be not active are getting activated because this repressive machinery is just not working perfectly anymore. And what we showed in this paper is that if you have a young data point where everything is still working properly, and you just artificially introduce noise to the system. So really artificially, we just add noise random numbers. And we do this, let's say 1 to 100 times. Then of course, just adding noise once still is somewhat resembling the original sample, but adding noise 100 times makes the sample, of course, much more divergent. So it's much noisier and not resembling the original sample at all. But what we do see is that aging clocks essentially linearly correlate with the amount of noise we added to the data. So we do see that clocks essentially just measure how noisy something gets. And I think this is really the crucial thing that we wanted to explain there, is that what aging clocks measure is not specific pathways that are going up or down, but just how noisy something is. So the pathways that are getting captured, like for example developmental pathways, are just a readout of, let's say, this imperfect maintenance system or how noisy your system gets. And it's just most easily visible in developmental pathways because these are mostly repressed. So if you have something that is, let's say, not completely repressed, the effect size is just a bit smaller, although you also can see that. And I think there's something important to understand, because it changed a bit of the story, that it's not like a continuation of developmental programs or maybe even a program aspect, but it's just how well your maintenance system works and how well it copes with the damage. And it's never perfect, so you just accumulate these damages. And this is then what these clocks seem to measure.
Your argument makes a lot of sense. And yeah, I read your paper that came out last year, and I'm glad that you've given me a better understanding of what you showed in that paper. But I think in the case of DNA methylation, I can understand that it maybe is just a reflection of this random noise that accumulates in cells of age. But regarding more like transcriptomic clocks, and I know that you previously published the transcriptomic clock, the bit age clock. Mm-hmm. And in that sense, I mean, of course, with transcriptome-based clocks, we have more information because we can see which genes are going up and down. And there's already like the classical, inflammation genes go up with age, or like there's some sort of predicted genes that we might expect in young versus old data sets. So how does that factor into what you're saying, that also there is just random noise in the data sets? And then also, if there's more noise with age, surely the aged cells themselves are more noisy, and therefore we cannot trust that data set so much. So how does that? Yeah, I think my brain's still a bit confused.
Yeah, there's a lot to unpack here. So regarding the transcriptomic clocks, you're right that there is, I think, more information in there. And maybe also one point that I should make clear is that it's not everything is noise, but there are also, for example, adaptive processes then. For example, if you have DNA damage appearing, so stochastic damage, of course you then have a program aspect afterwards, which is, for example, DNA repair or inflammation. So these are pathways that are also going up with age and inflammation essentially, especially, but these are adapting then to what happened before. So you first have DNA damage or something similar, and then in the response to that, you have inflammation also increasing. Which of course is then also important if you talk about correlation versus causation, because not everything that goes up with age is necessarily bad. Sometimes this is necessary to actually cope with the damage. So if you would reduce it, you would actually hinder the cell's survival and potentially actually reduce lifespan instead of increasing it. So it's a bit tricky to interpret that. But now with the pathways and so on that go up, that was also initially actually the idea why I wanted to build this C. elegans bit age clock that you mentioned. Because C. elegans is a really nice system for this. It doesn't live so long, so you can easily check pathways, genes, and so on, whether these do actually impact lifespan of the worms, whether they do impact fitness. And we have a lot of genetic and condition data sets available that allow you to really nicely directly predict the biological age and not the chronological age directly. So it's a bit easier to work with that compared to, for example, mammalian species. And we do see interesting pathways popping up in this clock. So we have, for example, a lot of systemic responses like the innate immune system, neuropeptides, and also a bit of DNA repair that does seem to play a role in the predictability of this clock. However, what we in the publication last year saw is that also transcriptomic clocks do correlate essentially linearly with accumulating noise in the data. It's not as clear as with an methylation clocks. I think there the signal is much clearer, so it's easier to build these noise clocks. With transcriptomic data, it's harder, but you still see that it essentially correlates with chronological age and also these biological age differences. So long-lived species and short-lived species are accordingly predicted, for example. And what we do think is happening there is something similar to what I mentioned before, is that if you have, in a young animal, you have all these genes repressed that are not supposed to be active, for example, also transposable elements. With age, the repression is getting weaker and you get an increase in the expression of these genes. And this is also part of the signal that is then getting picked up by transcriptomic clocks. So also this increase in noise seems to be also be relevant there. So it seems to be something universal. So at least everything we tested so far is behaving as we would expect, sometimes better, sometimes a bit worse. But all of the clocks that we tested do seem to track this accumulation of noise, even transcriptomic ones.
I see. And so it seems like it is, as you say, a combination of both noise and then also, as you said, that response to the noise, which is the DNA damage and trying to repair that damage. And so I think that somewhat nicely segues into a previous paper from your lab that you were part of, whereby your lab discovered the dream complex, which is a complex that seems to get repressed with age, which then sort of factors into some more suggestive of programmed aging. But anyway, to summarize, this dream complex is suppressing, in my mind, it suppresses the DNA repair machinery. Is that, maybe I got this backwards, but yeah,
That's
please
correct,
explain.
Yeah. Exactly. So the dream complex is a repressor. So it binds to genes and represses them. And it's mostly binding to cell cycle or DNA repressions and essentially, downregulates them. And indeed, as you mentioned, especially somatic cells have more active dream. So these cells repress DNA repair more. And as we have shown in this paper, is that if you reduce dream's activity or maybe knock it out completely, then you have increase in DNA repair gene expression and also DNA repair capacity. So we focus in the beginning here on the nematode C. elegans again. And there, if you have a mutant of dream, these worms cope much better with a variety of damages like UV radiation, ionizing radiation, also chemical treatments. They just survive much better because no repair is upregulated again. And the same we also see then in, for example, human cells. And there we even have like chemical inhibitors of dream that are also getting somewhat used in certain diseases, like, for example, Down syndrome, where not for the DNA repair part, but where it also does seem to play a role. So it's potentially even something that you could try in human studies sometime in the future.
And so in this particular case, so the dream complex seems to be increasing with age and thereby repressing the DNA repair proteins. Is that correct?
So what we do see is a few things. So DNA repair genes are often downregulated with aging. That's true. So most probably, it hasn't been definitely shown, I think, but probably cause is that dream is repressing these genes. And interestingly also, if you look across different species, you see that longer species have usually more active DNA repair. So this seems to cope better with damage and also live longer. So there seems to be a lot of evidence that dream might be really a master regulator of also how much damage accumulates in the cell and a good indicator of lifespan, for example. So I think that is definitely something that warrants further investigation. Now, why DNA repair is getting downregulated with age, I think that's difficult, and we don't know for sure at the moment. One point could be also, here this increasing noise. So you have genes that are supposed to be active, of course, getting more and more repressed with age. That's often the case. But we can, of course, also not rule out that there are other factors at play. It's, of course, impossible to rule out that any programmed or programmatic aging aspect is existing. But I think at the moment the favor is still towards the the accumulation of damage and epimutations, for example. But yeah, I think there's still many things to discover here.
No, exactly. And I think, my next question, which you sort of addressed, was whether this dream complex is due to just noise or it's part of some sort of program or like a response to the noise. And yeah, I agree. It's so unclear exactly what is the case. And I think, I know that there are perhaps two sort of fields in the aging space where some people think that aging is programmed and others think it's purely stochastic. I think personally I'm somewhere in between, but I suppose from what we've discussed, perhaps you are also.
So yeah, I think most of aging can be explained by this accumulation of noise, but we cannot rule out that there's other aspects. And of course, you have these differences in species lifespan. So I think instead of saying it's completely random, often what people say it's like quasi-random or quasi-stochastic, because there are also certain areas or aspects in your genome that are more prone to damage, for example. One point you can see is that there's a difference in transcribed versus non-transcribed regions. You have different repair pathways happening there. Then, of course, you have heterochromatic and euchromatic regions that are also more prone to certain types of damages and not. So there are a lot of things that make this random process more or less happening in certain aspects of your genome. You also have this effects that, for example, especially long genes are just more probable to be hit by damage. So just because the gene is longer, these genes are getting downregulated with age because they just are more probable to accumulate damage. So it's a quasi-random aspect that is definitely playing a role here. And then, of course, you have this evolutionary aspect where some species just evolve better maintenance system, better repair systems, and then can cope much better with damage. But we do think that the damage, as I mentioned all the way in the beginning, is what comes first. So aging is a default state. And then species just evolve better and better repair and maintenance systems to cope better with that. And then, of course, be able to live out so much longer and reproduce at a later age, for example.
So if we follow then your argument, then the way that we can help extend human healthspan then is to somehow increase the DNA repair capacity.
I think that is the most promising aspect. Of course, if you have inflammation, if you have like increased inflammation with aging, this is definitely not helpful. So reducing inflammation, which is this response to prior damages, is helpful and will also potentially be beneficial for your lifespan, but it doesn't get rid of the initial cause that introduces, let's say, aging to the system. So of course it's not proven, but I really think that what's come first is this random damage, be it DNA damage, epimutations, mutations to the genome, but also RNA protein damage, like protein misfolding and so on, of course, also play a role. And this then in the end leads to all these problems. Now we have already really good maintenance systems. We can already live for decades without having much problems actually and age-related diseases. But if we really want to combat this, I think we either have to improve maintenance, for example, with something like the dream complex, where we improve DNA repair capacities all the way, or we need to be able to somehow remove this random noise that is already in the system, which is, of course, nothing too easy to do.
Exactly. But in terms of like current interventions that do extend lifespan/healthspan, such as like Rapamycin in the mouse studies and caloric restriction, how is that reflected in these DNA repair pathways or in your clocks that you've been generating?
So a few things here. So if you reduce, for example, metabolism, let's say with calorie restriction, which of course by far now the best lifespan extension treatment that we have, nothing beats that so far. There are potential several things that happen. So first of all, we of course slow down metabolism. So we slow down potentially the endogenous damage source. But also if you have calorie restriction, the cells go from production to more maintenance. So now these cells upregulate something like autophagy, like lysosomes, potentially also DNA repair systems to better recycle what is already there and remove potential toxic waste and damages that are in the cell, which of course is then beneficial in the long run. Similar with rapamycin, mTOR is of course also related to that if you go from growth to maintenance, this is just beneficial to the whole setup of your organism. And what we do also see, I mean, with general aging clocks, but also with these stochastic aging clocks, how we call them, is that also there we do see this impact. So if we have calorie restricted mice, for example, we do with normal aging clocks, we do see a reduced age, but also with the stochastic clocks, which just measure, as I mentioned before, how noisy your system gets. Also, these do show a reduced age. So it does seem that this accumulation of noise is modulable. So either you reduce the damage or you might increase the maintenance. So I think both ways, or maybe it's a mix of both that we actually do see.
I see, yeah. And regarding just the clocks again, do you see them, I mean, of course, they're very useful for research, but in terms of the applications, do you see them more important for being able to track, like test certain interventions to see if they're working, or do you think they're more important for us trying to really uncover the key ways of intervening in aging? As in, I think maybe we've already somewhat answered these questions, but I feel like there's many aspects or ways you can use aging clocks.
Yeah, that is something that I think is also discussed in the field. A few review articles that came out recently on this. And I think one really important step in the future will be that if aging clocks can be used as an endpoint in clinical trials. Probably the clocks are not there yet. We don't have sufficient validation of aging clocks to really be able to claim that if a person is 5 years predicted younger, then definitely this person is getting to live 5 years longer, for example. We don't have sufficient longitudinal data. We have a lot of evidence that this might be true. There's a lot of association studies with mortality and also diseases and so on. So there seems to be predictive power. But I think the field still needs to make a next step, especially also to generate more longitudinal data and just validate these clocks much more. But that will definitely then speed up the whole field, of course, if we could say that if we measure now not lifespan, but an aging clock at a certain time point, and we can already infer the impact of a certain intervention. Now, on the other hand, I think aging clocks are valuable for discovery science. And this is also how we recently use clocks. So, for example, one thing that we recently tested them on is to see whether different cell types age at the same rate or whether there might be differences. And this is hard to do without aging clocks. And here we also focus now again on this cell elegance nematode system because it's just much easier and the cells in the worm are well defined. They can easily be tracked and so on. And what we do see there, for example, is that unlike from what you maybe expect that every single cell is the same age as its chronological age, that we do see differences. Some neurons, for example, neuronal cell types age essentially twice as fast as other cell types. And if you now look at these neurons, the aging predictions do hold true because those neurons that are predicted older actually degenerate much earlier. So we can, at a young adult stage, for example, predict with aging clocks which neuronal cell types degenerate earlier than others. And we can use that information then, for example, to identify what pathways actually are important for this degenerative differences, and also to see whether we can find compounds that might actually counteract this. So I think there's a lot of interesting discovery science still to be done. On another study, for example, we used again the cell elegance system, where we saw that you can also track certain rejuvenation events, even in somatic cells. And then, of course, can use this information to really delve deep and see what is actually driving these rejuvenating events. But maybe I put up a few topics here. I'm not sure whether you want to maybe ask questions first.
No, definitely. You absolutely have triggered many questions in my brain. And firstly, just to go back to what you were saying about the rate of aging, I think so. Firstly, yeah, I have many personal interest in like how different tissues maybe age at different times, different cell types, as you now seem to be describing. But even before we get to that point, at the moment we've discussed how these clocks, we call these linear clocks, and we show that they correlate with either chronological age or biological age. But I believe, I mean, it has been shown in a few studies, and I think it was last year there was maybe one or two studies came out and showed the sort of the two sort of transition points where perhaps we age a bit faster all of a sudden, suggesting that aging, it's going in the same direction, but maybe there's certain phases where it happens suddenly more rapidly. And so if we then say, aging stochastic is not programmed, how do we rationalize this information?
Mm-hmm. Yeah, I know the paper that you're talking about, and I think that's a really interesting study. Maybe before I answer the questions, I would like to see that validated also in a different data set. So it is easy with these data sets, if you have a focus on one data set, that there are artifacts that might skew your results a bit. I think it's still really interesting and it could be true, but I really would like to see this independently validated. Now, speaking about how this could be, I think that's an interesting point, and there are potentially two things that might be happening. One thing is that for example, also the accumulation of stochastic variation, the noise, that shouldn't be completely linear, because if you think about it, in the beginning everything is regulated nicely, and then you introduce noise to the system, and it's of course randomly distributed. But at some point, this noise will also hit maintenance factors, DNA repair factors, which then of course hinders maintenance and leads to an even faster accumulation of this noise. So in theory, it should speed up, and it should lead to an increase of this noise with age. So it could be that at some point you just see that happening. And then also, I mean, in the geophysics field, you have this idea that there might be certain thresholds. So maybe the cell is able to cope with a certain amount of damage, but then once a certain threshold is reached, this really speeds up. You can also, if you think about like a specific gene that is repressed by DNA methylation, for example, there is potentially a certain buffer mechanism at play. So, for example, if you have a gene with, let's say, 5 CpG DNA methylation sites that repress the gene, and everything is methylated, so the gene is repressed, it can cope with some kind of error. So, if one of the 5 DNA methylation flips, you have still 4 methylated, so the gene is probably still pretty well repressed. Of course, with time, you might have additional epi mutations, and the gene is getting expressed again slowly. But you might, of course, have differences now in the length of the CpG sites. Some genes or species also might have more or less, leading to more or less buffering mechanisms, which could explain some of these thresholds that are hypothesized in the geophysics field. And I think there's also some evidence that this is what might be happening. So it could be that at these time points where they see this abrupt changes in the speed of aging, that is what is happening. And then, of course, you also have some parts that are regulated. For example, in women, you have menopause, where you have, of course, a dramatic change in general biology, which, of course, is hormonally regulated, which might, of course, also lead to a difference in the speed of aging before and afterwards. So I think there are several things at play that we don't completely understand yet. But I think it will be interesting to delve deeper into these kind of turning points to see how we could explain them. Completely agree.
No, absolutely. I think you're right. There probably are some very complex mechanisms at play. And perhaps just on average, if there's many genes with these sort of thresholds, then on average there is sort of a linear by DNA methylation or other clocks. Whereas I believe they use proteomics, right, to see the different change points, but I can't quite remember what they showed in that paper. But the other topic I wanted to touch upon was people are also using these aging clocks, to actually show how cellular reprogramming, going from a differentiated cell back to a pluripotent cell, also seems to somehow rejuvenate cells. And part of my brain just can never fully understand how exactly that works, given that we think aging is a stochastic process. What exactly is happening potentially in these cells to erase these random marks that have arisen with age?
I think there are two things at play here. First of all, with reprogramming, we have cell removal, so especially damaged cells potentially just die. So if they can't cope with reprogramming because they are too damaged, they will of course then just get removed. So the, biologically speaking, younger cells are then retained. Then of course you also have cell division, which dilutes damage. So if you have protein damage, RNA damage, or also potentially DNA damage, and you replicate, this is of course diluted, so you have a certain effect there. But then also what I think is happening is if you have this reprogramming, what it's doing is essentially a factory resetting. So the cell's epigenetic makeup, at least, is really reset to what it was at the beginning. And this is essentially also what is happening in embryogenesis, right? You have two old cells, the egg and the sperm, and after they come together, these two old cells somehow rejuvenate to something that can give rise to a completely new organism without damage, maybe except mutations, but of course are not really repairable. But really showing that this epigenetic information might be really the most important point, that you, if you can reset this and get rid of this noise in the system, that is then actually really helping the cell to rejuvenate.
And I think that actually does make a lot of sense. And so in that case, of course, there's many companies now at the moment and other research labs that are trying to find ways to rejuvenate cells by this epigenetic reprogramming. And so therefore, do you think that this is like a logical approach for cell rejuvenation?
I think it's one of the most promising approaches. Of course, there are many challenges that we still have to overcome. We don't want to reprogram everything to iPSCs, for example. That, of course, wouldn't be healthy. But also this partial reprogramming seems to work reasonably well. And I think that could be really one of the most promising aspects to get rid of this noise. I mean, I know there are some people saying that you cannot get rid of all the entropy in the cell. Some people cite like the second law of thermodynamics, that if entropy increases, you just cannot get rid of it. But I also would like to make a point here that I don't think that is true, because I mean, entropy increases in closed systems, but your body is an open system. And what we do every day is essentially we consume energy and we get rid of waste to somehow reduce the amount of entropy that our cells have to cope with, right? So there are mechanisms at play that can remove this stochastic damage. And I think OSKM reprogramming or similar partial reprogramming approaches are really just a culmination of this, and really the way to really get rid of most of the entropic damage. How exactly it's getting done is, of course, still difficult to answer. I think it's this combination of the things that I mentioned before, but I think it's a good approach for the future.
Exactly, and I think the same, and of course that's probably why there's so many people interested in it and studying on it at the moment. But I guess I'm more curious to know what other things perhaps you're reading about in the aging field, or other aspects that you think are currently maybe underrated or overlooked in the aging space.
Underrated things in the aging space. That's a good question. One thing that I'm personally quite excited about, and I think it's not studied too much at the moment, is the rejuvenation events that are happening in post-mitotic cells. So as I mentioned before, if you have reprogramming, you have this usually in cycling cells. You have damage dilution, you have apoptosis and so happening. And this is, of course, something different from what would happen in a post-mitotic cell with cannot replicate anymore, like your neurons, for example. You usually don't have replication there anymore. So how would that then work in neurons? And there's not much research done on that. I mean, it's, of course, harder to study. But one thing I briefly mentioned before is a study that we are looking at the moment is the diapause state in C. elegans. So C. elegans has several developmental stages. It's quite complicated. And then in the end, it reaches adulthood. And if you want to study aging, you usually only look at adult worms to not confront yourself with all these differences that happen during development. But now, if C. elegans is on a sparse diet, or if it's overcrowded, then these worms can enter into a diapause, or in fact, several diapause states. The most well-studied one is called Dauer, and they can live in this diapause state for much longer than they would live during adulthood. They slow down metabolism, they still move, but they don't die for essentially months. And once they exit this diapause, they essentially live normal lives. They have nothing is wrong with them. They have normal lifespans, they have normal progeny, there's just nothing wrong. And the question here is really, how is that possible? There are potentially two different things happening. One is that they just don't age during this diapause stage, which is probably not happening because they really are in this state for weeks or months. And the other is that there is a rejuvenating event upon exit of this diapause state, which must be completely happening in post-mitotic cells because these worms are already essentially developed. So except the germline, which then develops afterwards properly, these worms are post-mitotic. So there cannot be damage dilution and there cannot be apoptosis happening. That is not what's rejuvenating these worms. So it seems to be something cell intrinsic that is important for this rejuvenating effect. And what we do see in these worms is that they indeed do age. So if we apply, for example, aging clocks, we do see an increased age over the time they spent in Dauer. And then amazingly, once they exit, they do actually rejuvenate, at least transcriptionally speaking, which I would say makes a lot of sense because you, of course, also accumulate metabolic damage and byproducts over the time spent in this diapause. And I think this is really something that is not getting studied enough at the moment to how this is possible. How can there be rejuvenation events happening in a post-mitotic organism? And I think that's really something that people should also focus on more. I think there's a lot of things that could be really beneficial for the whole field if you could delve deeper.
No, I completely agree because also, I mean, regarding translating that to human therapeutics, if we want to find ways to intervene later in adulthood, our cells are already damaged. I mean, we discussed probably mainly about the DNA methylation and with epigenetic reprogramming, how we can rejuvenate cells, but we're mainly just changing DNA methylation marks. We're not necessarily fixing the mutations that are still present in the genome. So I think if there are ways to rejuvenate cells, ignoring the fact that maybe there are mutations that we don't maybe have to deal with them, then I agree that it's very interesting and probably that's cool that you found a system that you can study that in.
Yeah, I think it seems really promising. I think it's a good system. There are also other diapause states that do seem to behave similar. So for example, in a collaboration with Adam Atebi, where they look at an adult diapause state, they essentially do see the same thing happening. So they do age and then they rejuvenate upon refeeding. And I think if we can understand this better, we can really find also novel potential treatments that are maybe not as easy to work with in humans, but I think necessary. We of course also have post-mitotic cells in humans, like neurons, where we potentially cannot use easily these treatments that might rely on damage dilution or also apoptosis of damaged cells. So I think these approaches are really necessary also in the field to move forward.
Yeah, absolutely. And yeah, speaking of the aging field in general, where do you see the field sort of going in the next few years, or like, are you optimistic, or how do you feel about the way things are going in terms of research topics, etc.?
I think it's really picking up speed. There are a lot of interesting studies coming out recently, also more funding getting into the field, which is fantastic. The aging clock field still has a lot of challenges ahead, I think. One problem being, which I briefly touched upon before, is that we don't have a lot of validation or longitudinal data available. So this is definitely something where a focus will be in the future. But generally also the aging field we get a much better understanding now on the different aspects that might drive aging. I mean, this accumulation of noise is one thing, but of course we also need to understand the adaptive response better. Or also there are now more and more cross-species comparisons, which I think are really important to understand what pathways and genes are specifically important for evolution of longer lifespans. DNA repair is most of the times popping up, so usually you find mutations or increase in DNA repair genes in longer lived species, but these studies are really still rare now. So I think these cross-species comparisons really will be important, and people start picking up on that, and I think this is really exciting.
Yeah, no, I completely agree. I think there's still, unfortunately, still so many things we don't understand, but that also does mean there's something for us to look into as researchers. So yeah, I can only wish you the best of luck with your continuing studies. Yeah, it's been great to hear about your research today, and yeah, I hope to hear or read more about your work in the future.
Yeah, thank you for having me here. It was really fun talking to you.